I am Dr. R. Krishnan, Assistant Professor, PG and Research Department Microbiology, St.Joseph College of Arts and Science, Autonomous Cuddalore. Today, I am going to present a lecture, a PPT, regarding agglutination and precipitation reactions. So, what is agglutination? A definition. So, agglutination is the process of clumping of antigens with their respective antibodies. Agglutination reactions are used to particulate test antigen that are usually conjugated to a carrier. The carrier can either be artificial, such as a latex or a charcoal particle, or it may be a biological molecule, such as red blood cells. The agglutination process involving red blood cells are termed heme agglutination, and the process in which white blood cells is termed leukoagglutination. These conjugated particles then react with the patient's serum, which might or might not contain antibodies. The result of this test can be collected based on the observation of clumps resulting from that antigen antibody complex formation. The sensitivity and accuracy of the result depends on a number of factors like the duration of incubation with that antibody, amount and strength of the antigen conjugated to the carrier, and the test condition like the pH and protein concentration. Different forms of agglutinations are used in diagnostic tests, so which include latex agglutination, flocculation test, direct bacterial conjugation or agglutination, and heme agglutination. Because the process is quick and straightforward, this diagnostic tests are preferred over other sophisticated tests. Agglutination has been used for the detection of antigens in bacteria, which ultimately helps in the identification of those bacteria. A modified and more sensitive technique associated with the agglutination is agglutination PCR. The agglutination formed is then used to quantify the DNA strands by qPCR method. So next is precipitation. The definition is precipitation is a process where soluble antigen bind with their specific antibody at an optimum temperature and pH, resulting in the formation of an insoluble precipitate. The interaction between the soluble antigen and antibody results in the formation of insoluble lattice that forms precipitate out of the solution. The process of precipitation has some requirements involving the valency of both antigen and the antibody. For precipitation to take place, the antibody must be bivalent and the antigen must be either bivalent or polyvalent. Precipitation takes place in a zone of equivalence where the concentration of antigen and antibody is equal. On the other side of equivalence, precipitation does not occur if the concentration of either antigen or antibody is in excess or deficient. So next is immunological techniques like immunodiffusion and electroimmunodiffusion utilize the principle of precipitation reactions. The principle of precipitation is also applied in analytical chemistry for the detection of various functional groups of chemical substances. If a lighter participate is formed, another process termed flocculation takes place. In flocculation, the precipitate floats instead of sedimenting. Precipitation reactions are commonly performed on a semi-solid surface like agar media or non-gel support media like cellulose acetate. The precipitate formed in the reaction remains suspended until enough force of gravity can settle the precipitate towards the bottom of the surface. So next is some of the examples of an agglutination reactions. So one is called hemagglutination assay. So hemagglutination assay is a diagnostic technique used for the detection of viruses, bacteria, and antibodies. The antigens present in various viruses or bacteria bind with the sialic acid receptors present on the surface of red blood cells, so creating a network of RBCs and viral particles. The formation of these lattice particles depends on the concentration of virus, bacteria, and RBCs. When the concentration of the antigen is too low, RBCs are not arranged in lattices but settle down at the bottom of the container. Hemagglutination is based on the same principle as the one used by virus during infection. Second example of an immunodiffusion is or precipitation is immunodiffusion, a precipitation test. Immunodiffusion is an immunological technique used for the detection and quantification of antibodies and antigens, so which are mostly immunoglobulins and nuclear antigens. In this technique, antigen and antibodies are applied simultaneously in two adjacent wells. As the antigen and antibody diffuse towards each other, precipitates are seen in the form of lines as the antigen and antibodies interact with each other. It is also possible to compare the concentration of different antigen by placing multiple antigen in multiple wells. So based on the formation of precipitin line, the presence of different antigens and in turn the presence of virus or bacteria can be detected. So this is the image which actually shows the difference between agglutination and then precipitation reactions. In first cases, where the antibody binds to the surface of the antigen on the bacteria. In second example for precipitation, so where the suspension containing the precipitate due to the formation of antigen antibody complexes at the bottom. Thank you.